Alright, hello everybody. In this presentation, I will be discussing chapter 24 of the cell cycle and mitosis, particularly section 24.3, the regulation of the cell cycle. So to begin with, this is the key transition point in the cell cycle. The red bars indicate the three main transition points that are presented in a eukaryotic cell cycle. They include one at the end of the G1 phase with a restriction point that allows cells to enter the S phase, and it is monitored by the growth factors, amount of nutrients, the cell size, and if the DNA is damaged or not. The second transition point is at the end of the G2, which is the G2M phase, and it is monitored by the cell size, if the DNA is damaged or not, and the number of DNA replication it has undergone. The third transition point is at the end of the M phase, in which the metaphase anaphase transition that is monitored by the chromosomes if they are attached to the spindle or not. And then the dark green arrows are late G1 and G2 locations where the cell can exit from the cycle and it can enter a non-dividing or arrested state. So this slide shows the role of chemical signals in cell cycle regulation. So figure A showcases that when a cell is in S phase, is fused with the cell that is in G1 phase, the G1 phase cell is then activated to the S phase and DNA synthesis begins. And figure B showcases that when a cell is in the M phase, when fused together with the cell in G1 or any other phase than M, that cell will then be activated to the M phase and the chromosomes duplicate. So this showcases that molecules that are in the cytoplasm push cells from G1 to S phase and from G2 to M phase. So here we have the fluctuating levels of cyclins and MPFO activities during the cell cycle. In the cell cycle, a CDK cyclin complex controls the cell cycle. It is managed so that it only functions at the end of G2 phase. It is managed by keeping the concentration of the meiotic CDK constant and the concentration of the mitotic cyclin limited so that it is not present in the cell all the time. And CDK is only active when attached to mitotic cyclin. And so figure A showcases the level of M-phase cyclin during the cell cycle. So you can see the gradual increase of mitotic cyclin in the G1 S phase and then meets a threshold at the end of the G2 phase that binds to meiotic CDK to form the CDK cyclin complex and start mitosis. Then halfway through mitosis, cyclin is then destroyed and the CDK activity decreases, which prevents another round of mitosis to occur. And then in figure B showcases the levels of different cyclins during the cell cycle. The original mitotic cyclin is cyclin B, and the rest of the cyclins undergo changes based on it. And so the availability of these different cyclins to form complexes with various CDK manages important phases in the cell cycle. So this slide goes over the regulation of mitotic CDK cyclin by phosphorylation and dephosphorylation. And the CDK cyclin complex requires the phosphorylation of mitotic CDK in order to become activated. And so as seen in the figure, CDK cyclin is initially inactive, and whenever two inhibiting kinases binds to the CDK molecule, it blocks the active site. And so the activating phosphate group will then add to a specific activating kinase location that causes the inhibiting kinase to be removed and the CDK cyclin complex to be activated. So this is a slide of the mitotic CDK cycle. And so once the mitotic CDK cyclin is activated, its protein kinase activity begins mitosis that causes the nuclear envelope to break down by phosphorylating the nuclear lamina that depolymerizes the lamina protein, causing the lamina to break and the envelope to be torn apart. And the chromosomes also start to condense and become visible, and the mitotic spindles begin to form, and the targeted protein cyclin is then targeted and gets broken down and degraded. So this is the anaphase promoting complex, and the CDK cyclin not only plays a role in the start of mitosis, but it also plays an important role later in mitosis when the separation of sister chromatids is required for anaphase. And so the APC, or the anaphase promoting complex, activates an enzyme ubiquitin ligase that breaks down securin, which is a protein that keeps sister chromatids from coming apart. And so before anaphase, sister chromatids are held together by a cohesion protein that is monitored by securin. And it makes sure that the sister chromatids are attached before separation to opposite ends by blocking the protease separase that breaks down cohesion protein, causing sister chromatid separation. And so the anaphase promoting complex is activated. Once it is activated, it breaks down securin, which allows sister chromatids to separate and allows mitotic cyclin to be degraded, which causes cytokinesis and the decondensation of chromosomes and the reassembly of the nuclear envelope. So this is the role of RB protein in cell cycle control. And so the RB protein is initially bounded to the E2F transcription factor in its inactive form, which inhibits gene transcription. And so the phosphorylation of RB protein by G1 CDK cyclin activates RB protein, and it releases it from the E2F transcription factor, which causes E2F to become activated and begin gene transcription that is needed for the S phase entry. So this is licensing of DNA replication during the eukaryotic cell cycle. And so the transition to S phase from G1 is a key point in the cell cycle. And it confirms that a cell's nuclear DNA that has entered the S phase for replication only divides once before cell division. And so licensing begins when the ORC and the helicase loaders help MCM proteins bind to the DNA at the origin of replication. And so then CDK and geminin proteins are made to block the ORC and helicase loaders from binding to MCM, which does not allow for MCM to then bind to the origin of DNA replication. The origin of replication on the DNA, and that stops relicensing. So you can see that once the S phase begins and until the M phase is complete, licensing is stopped by the CDK and geminin proteins. So this is the spindle assembly checkpoint, and the spindle assembly checkpoint is in the metaphase anaphase during mitosis. And it does not allow for chromosomes to be pulled apart before they are connected to the spindles. And so figure A showcases that the kinetochore and spindle microtubules that are not connected will start to build up proteins of MAD and BUD families that are also part of the mitotic checkpoint complex that produces a weight signal that stops the anaphase promoting complex from binding to the CDC20 protein, and the CDC20 protein is what turns on the anaphase promoting complex that causes the sister chromatids to be separated during anaphase. And so figure B showcases the fluorescence of a cultured Drosophila cell that formed BUB1, which is a red dot at the kinetochore of the chromosome because the chromosome was too slow to get to the metaphase plate, which is the arrow shown in the figure, when treated with the low doses of pilotaxel. So this is, lastly, let's go over the role of p53 protein in responding to DNA damage. And so the last checkpoint is important in that it stops and prevents damaged DNA cells from going through the cell cycle unless they get fixed first. And so that occurs multiple times in the cell cycle, like in late G1 phase and in the S phase and in late G2 phase by stopping the CDK cyclin complex. And so the protein p53 plays a central role in this event by activating other proteins and enzymes that prevent cell division of damaged DNA cells from occurring. And so that is why p53 is also referred to as the guardian of the genome. And so p53 is a protein that needs to be activated in order to work. And so when p53 is bounded to the MDM2 complex, it is inactive and is beginning to be degraded. But whenever the checkpoint kinase phosphorylates p53, it gets released from the MDM2 complex and it becomes activated and proceeds to activate other pathways. And so p53 can activate two different pathways, and the first one is the cell cycle arrest pathway, and that is where activated p53 will activate a protein p21 that stops CDK from binding to cyclin, which forms a CDK cyclin complex, which then goes and activates the RB protein and promotes gene transcription, as we saw earlier. And so since p21 blocks CDK, gene transcription cannot occur and the cell cycle is stopped. And so the second pathway p53 can activate is the apoptosis, which is programmed cell death pathway. And that is where activated p53 will activate a protein called PUMA that stops the function of the protein BCL2, whose main function is to not allow apoptosis to happen. And so because PUMA blocks BCL2, apoptosis is not stopped, and so the cell death of a damaged DNA cell is then programmed, and it leads to apoptosis. And so here are my resources and my references, and I hope that you all learned something. Thank you.